Hello everyone. So in the previous part we have discussed that types of G protein the structure of G protein coupled receptor, types of G alpha subunits, and but in this part we have discussed about the G alpha stimulatory subunits, that is the signaling of G alpha stimulatory subunits, and this can be understood by the examples: regulation of glycogen metabolism by epinephrine. So how glycogen metabolism is regulated by hormonal hormones, that is hormone mediated glycogenolysis. So here hormone is epinephrine. So in case of low blood glucose level, epinephrine kinase secretes epinephrine that activates epinephrine, and this leads to the stimulation of glycogen breakdown, and glycogen synthesis is stimulated. So the mechanism involves, here you can see this is epinephrine signals, and those signals are bind to the GPCR epinephrine receptor, and in the C terminal, as I have told before, that in the C terminal there is G alpha subunit, stimulatory subunit, along with gamma and beta, and it is attached with the GDB, that is in inactive form, and then after signaling it is attached with GTP and becomes active. So G alpha stimulatory subunits, when activated, it leads to the formation of CAMP from ATP, and this helps in adenylyl cyclase. So what is adenylyl cyclase? Adenylyl cyclase is an enzymes that helps in the conversion of ATP from cyclic AMP, and adenylyl cyclase are of two types. One is transmembrane adenylyl cyclase, and another is soluble adenylyl cyclase. So transmembrane adenylyl cyclase is found in plasma membrane, whereas soluble adenylyl cyclase is found in cytosol. This CAMP is also called secondary messengers. Why it is called secondary messengers? So there is epinephrine signaling, that is fast messengers, and the CAMP is called secondary messengers. So secondary messengers are those molecules that small molecules or ions that relay signals from the cell surface receptors, that is cell surface receptors, and to the target proteins. So here you can see that epinephrine is a signaling molecule that relays signals through cell surface receptors, and then the effector proteins are activated through CMP. That is why CMP is called secondary messengers, and when it binds to the protein kinase A, that has two subunits, that is regulatory subunits and catalytic subunits. So protein kinase A is a proteins that is an enzymes that phosphorylate proteins, that is located in the it is located in the cytoplasm. So protein kinase A is also known as CAMP dependent protein kinase, as its activity is depends on the cellular levels of cyclic AMP, and protein kinase A is an heterotetrameric. Here you can see the tetrameric structures that is consist of two regulatory subunit and two catalytic subunits. So catalytic domains are generally masked or covered by the regulatory subunits. Catalytic domains are covered by regulatory subunits and prevent the kinase activity of this protein kinase A, and in the absence of CMP. But when CMP binds to this protein kinase A, the regulatory subunits are removed, dissociated, and then the catalytic subunits are activated. So here you can see that protein kinase A is activated and regulatory subunits are dissociated. Now come to the this part. There is a glycogen synthase that is in active form, and glycogen synthase uh, causes synthesis of glycogen molecules. So if there is synthesis of glycogen molecules, that means there is already a lower blood glucose levels lowers. So if glycogen synthesis molecules is activated, then the blood glucose levels lowers more. That's why protein kinase A activation leads to the inactivation of glycogen synthesis by adding of one phosphorus molecule, and then glycogen synthesis is inhibited. And on the other hand, protein kinase A activates glycogen phosphorylase kinase, inactivated glycogen phosphorylase kinase to active glycogen phosphorylase kinase by adding one phosphorus molecules. And there you can see that glycogen phosphorylase again activates the glycogen phosphorylase by adding one phosphorus group, and there glycogen breakdown occurs, which leads to the high glucose level in blood. And there these activated catalytic subunits enters into the nucleus, and there you can see the protein kinase catalytic subunits activated, and it binds with the crab protein. Now what is this crab protein? This crab protein is an intracellular protein that regulates the expression of genes, and it binds in the CRE side, that is CAMP response element. So crab protein is CAMP response element binding protein. So it is bind in the CRE side, that is CAMP response element, and there is a HAT coactivator that activates the binding, and target gene is expressed. So in the epinephrine mediated glycogenolysis, this occurs, and thus blood glucose level is maintained by epinephrine. So these epinephrine signal stimulate first G alpha stimulatory subunits and activate beta androgenic receptor. There is, you can see there, GPCR epinephrine receptor, which is a beta androgenic receptors, and this leads to the increasing heart rate, and it occurs when flight, fight, or fright. There's three type of responses. This epinephrine signal has an agonist and antagonist. There is a role in of agonist and antagonist in this mechanism. So what is agonist? Agonist behaves like a signal, and it can activate same pathway like signaling molecule. Agonist like this signaling molecule, and like this signaling molecule, agonist can activate the same pathway and it behaves like signals, whereas antagonists are inhibit the signal molecule functions and block the signaling molecule receptor. That means it is a opposite of agonist. Antagonist is the opposite of agonist. Agonist activates the pathway, whereas in antagonist inhibits the pathway, signaling pathway. So that's why antagonist is the opposite of agonist. It blocks the signaling molecule receptor. Antagonist. So examples of agonists are aspartame, saccharin, monosodium glutamate, whereas antagonists are beta blockers. So beta blockers are the antagonists of epinephrine. Here, beta blockers block the signaling molecule and block the role of epinephrine signaling. So if epinephrine signaling is not occur, then those whole mechanism does not occur. If epinephrine signaling is not occurs, then the whole mechanism does not occurs. So this is the role of agonists and antagonists, and thus G alpha stimulatory subunits are regulated, are activated, and the epinephrine mediated glycogenolysis are regulated. In the next part, we will discuss about the G alpha inhibitory subunit and its signaling pathway.